All right. So remember, at the end of last class, we were talking about the innate or non-specific arm of the immune system. We talked about barriers to the entry of pathogenic microbes, and that was sort of where we left off. So I'm not going to review the barriers to entry. You can look through your notes for that. I'm just going to pick up with inflammatory response. All right, so if a pathogen gets past all of the preventative mechanisms, so the second line of defense limits the spread of pathogens, sort of localizes any infection, removes any waste products, and begins the process of tissue repair. Inflammatory response is non-specific in the way that it deals with pathogens. All right, so when tissue damage occurs, immune cells that are called mast cells, these are the resident tissue immune system cells that I was telling you about. They release a molecule called histamine, and histamine causes the capillary beds in the area to dilate, right? So it increases blood flow to the area, and it also makes the capillaries leakier. That allows for the movement, or easier movement of white blood cells, such as neutrophils and monocytes, to move out of the bloodstream and into the tissue. Histamines are an example of a kind of molecule called a cytokine. It's essentially a local signaling molecule. Cells that are damaged also release molecules called chemokines. And these are very similar to cytokines. The difference is essentially that chemokines are used by white blood cells to find the source of the infection. So it's a longer distance signaling. And then finally, you have neutrophils, monocytes that have differentiated into macrophages, phagocytosing the pathogens. Right, so in the cartoon here, we have some pathogens, let's say on the surface of the skin. There's a break in the skin that allows pathogens in, and that starts the inflammatory reaction. You can see in the bottom panel, there is an increase in a swelling in the tissue that's related to additional tissue fluid, interstitial fluid, that's generated as the capillaries become more permeable. This slide is just showing you, it's a reminder about how phagocytosis works. So you want to remember it's active transport, which means that it requires the cell to invest its own energy in the form of ATP. What we're not seeing in this image is all of the reorganization of the cytoskeleton that's used to create these pseudopodia, which is Latin for false legs. Those are pseudopods right there. They rise up and around the pathogen, create a vacuole or vesicle, and then a lysosome, which is full of digestive enzymes, fuses with the vacuole, creating a phagolysosome. In many cases, the next step of the process is that once say we're talking about a bacterium, once it's been killed and digested, some of the parts of the bacterium are embedded into the phagocyte's cell membrane, and they can then present those molecules, which are called antigens, so that the adaptive immune response can take over. Neutrophils are the first to leave the capillaries. And remember from class the other day that neutrophils are the most common kind of white blood cell, and they are fantastic at phagocytosis. What I want you guys to take away from this slide is that there are 4 hallmark symptoms of inflammation: heat, redness, swelling, and pain. All of those are mediated by cytokines and chemokines. So capillary widening leads to increased blood flow. That accounts for the additional heat because blood, remember, is at 100.4 degrees Fahrenheit, and skin temperature is lower than that. That increased blood flow is also going to lead to redness. The increased permeability mediated by the cytokines is going to lead to increased fluid in the tissues, which leads and then increased attraction of white blood cells, all of which are going to increase the volume of that tissue, causing swelling. And swelling is going to push on nerve endings that aren't normally stimulated, and that generates pain. When you have a fever as a result of infection, it's when the inflammatory process has moved to the next level. So it's not a local response any longer. Right? Remember when we talked about the nervous system, that the hypothalamus is a part of the brain that's responsible for controlling a lot of vegetative functions. The hypothalamus stimulates the body to produce molecules called prostaglandins, and those, through a set of molecular cascades, leads to additional molecules that trigger fever. Prostaglandins are molecules that are involved not just in fever, but they're also associated with pain. Fever is, in general, fever is an adaptive response by your body to the presence of pathogens. Excuse me. Pathogens are quite happy at our typical body temperature, but when that temperature goes up, they are far less likely to survive, and that gives the immune system a leg up. At a certain point, though, fever becomes problematic for our own cells, because remember that proteins change shape when you change temperature too radically, and our enzymes will stop working. Okay, so also considered part of the second line of defense of our immune system is an array of protective proteins, including the complement system, and which is present in blood plasma, as well as the interferons. Complement is called complement because those proteins complement the function of the immune system. They're actually made in the liver and then pushed out into the blood. And there are a wide variety of different mechanisms by which they help the immune system. One of my favorites is shown in the cartoon in this slide. The complement proteins that produce membrane attack complex. So when these proteins encounter a bacterial cell, they sit down on the surface of the cell and arrange themselves just so that they create a hole in the cell. Now remember that tissue fluids are going to be hypotonic to the interior of the cell, and when you have a huge opening in the cell, like you have when you have a membrane attack complex in play, you end up with fluid and ions rushing in, and the bacterial cell explodes. Other kinds of complement proteins can bind to mast cells, those resident immune system cells present in tissues, to stimulate the release of histamine, so they influence the inflammatory response. Another category of protective proteins are called interferons. And interferons interfere with the ability of viruses to infect our cells. Interferons are only produced by a cell when it's been infected with a virus. And it's essentially a warning call. So interferons are produced, they're exocytosed. Neighboring cells have receptors that can bind the interferon, and that's a signaling mechanism that tells the uninfected cells to start sort of battening down the hatches, so build additional protective proteins to keep themselves from becoming infected. The production of interferons does not help, as far as we can tell, does not help the virally infected cell. It's either going to die because it becomes so full of virus that it lyses, or because a cytotoxic or natural killer cell takes it out. But it does help all of the cells around it. So remember, when we're talking about any organism, we all have unique cell surface molecules. And our immune system learns how to tell self from non-self early in development. All right, so finally, we're going to talk about natural killer cells. They are in the from the lymphoid line, but they're different than cytotoxic T cells. Natural killer cells have a way of recognizing virally infected cells or tumor cells. So these are our own body cells that stop producing a protein called major histocompatibility complex with a specific self-antigen. So the natural killer cells recognize what's referred to as missing self. And the way they work is really remarkable. They come up next to one of our cells that's abnormal, and the natural killer cell actually rotates the Golgi body. Remember, the Golgi body is sort of the post office of the cell. It's responsible for packing molecules into vesicles. It rotates the Golgi body toward the abnormal cell and exocytoses a bunch of molecules called perforins. Perforate means to punch through. So these perforins self-assemble on the abnormal cell, creating holes in the cell membrane, and that leads to lysis of the abnormal cell. All right, so that's it for the first and second lines of defense in the immune system.